organisms retain the enzymatic machinery to partially oxidize glucose without the help of oxygen. This breakdown of glucose to pyruvic acid is called glycolysis. The term glycolysis has originated from the Greek words, glykos for sugar, and lysis for splitting. The scheme of glycolysis was given by Gustav Embden, Otto Meyerhoff, and J. Parners, and is often referred to as the EMP pathway. In anaerobic organisms, it is the only process in respiration. Glycolysis occurs in the cytoplasm of the cell and is present in all living organisms. In this process, glucose undergoes partial oxidation to form two molecules of pyruvic acid. In plants, this glucose is derived from sucrose, which is the end product of photosynthesis, or from storage carbohydrates. Sucrose is converted into glucose and fructose by the enzyme invertase, and these two monosaccharides readily enter the glycolytic pathway. Glucose and fructose are phosphorylated to give rise to glucose 6-phosphate by the activity of the enzyme hexokinase. This phosphorylated form of glucose then isomerizes to produce fructose 6-phosphate. Subsequent steps of metabolism of glucose and fructose are same. In glycolysis, a chain of 10 reactions, under the control of different enzymes, takes place to produce pyruvate from glucose. While studying the steps of glycolysis, please note the steps at which utilization or synthesis of ATP or, in this case, NADH and H+ take place. ATP is utilized at two steps. First in the conversion of glucose into glucose 6-phosphate and second in the conversion of fructose 6-phosphate to fructose 1,6-bisphosphate. The fructose 1,6-bisphosphate is split into dihydroxyacone phosphate and 3-phosphoglyceraldehyde, GL. We find that there is one step where NAD plus H plus is formed from NAD plus, this is when 3-phosphoglyceraldehyde, GL, is converted to 1,3-bisphosphoglycerate, BPGA. Two redox equivalents are removed, in the form of two hydrogen atoms, from GL and transferred to a molecule of NAD+. GL is oxidized and with inorganic phosphate to get converted into BPGA. The conversion of BPGA to 3-phosphoglyceric acid, PGA, is also an energy-yielding process. This energy is trapped by the formation of ATP. Another ATP is synthesized during the conversion of PEP to pyruvic acid. Can you then calculate how many ATP molecules are directly synthesized in this pathway from one glucose molecule? Pyruvic acid is then the key product of glycolysis. What is the metabolic fate of pyruvate? This depends on the cellular need. There are three major ways in which different cells handle pyruvic acid produced by glycolysis. These are lactic acid fermentation, alcoholic fermentation and aerobic respiration. Fermentation takes place under anaerobic conditions in many prokaryotes and unicellular eukaryotes. For the complete oxidation of glucose to CO2 and H2O, however, organisms adopt Krebs cycle, which is also called as aerobic respiration. This requires O2 supply. Fermentation. In fermentation, say by yeast, the incomplete oxidation of glucose is achieved under anaerobic conditions by sets of reactions where pyruvic acid is converted to CO2 and ethanol. The enzymes, pyruvic acid decarboxylase and alcohol dehydrogenase, catalyze these reactions. Other organisms like some bacteria produce lactic acid from pyruvic acid. In animal cells also, like muscles during exercise, when oxygen is inadequate for cellular respiration, pyruvic acid is reduced to lactic acid by lactate dehydrogenase. The reducing agent is NADH plus H+, which is reoxidized to NAD+ in both the processes. In both lactic acid and alcohol fermentation, not much energy is released. Less than 7% of the energy in glucose is released and not all of it is trapped as high-energy bonds of ATP. Also, the processes are hazardous, either acid or alcohol is produced. What is the net ATPs that is synthesized? Calculate how many ATP are synthesized and deduct the number of ATP utilized during glycolysis, when one molecule of glucose is fermented to alcohol or lactic acid. Yeasts poison themselves to death when the concentration of alcohol reaches about 13%. What then would be the maximum concentration of alcohol in beverages that are naturally fermented? How do you think alcoholic beverages of alcohol content greater than this concentration are obtained?